In chapter 5, we are going to move on from carbohydrates to lipids as we move through all of our essential nutrients. So our learning outcomes, and you can use this as a study guide to help for the next test. All right, what we'll be looking at here in the first section is just some of the chemistry, so the properties of lipids, the different types of lipids that relate to nutrition and what they do. So first of all, just what lipids are, so some of the common properties. Mainly what unifies them all is that they do not dissolve in water. So this is a very diverse group of compounds, and a lot of them don't even have anything to do with health or nutrition. So we'll just focus on the ones, of course, that do. But again, any molecule that does not like water is grouped into this big grouping of lipids. Of course, as we learned before, lipids do provide us the most amount of energy per gram, providing 9 kilocalories per gram. And then just some, kind of definitions or differentiations. Fats are going to be solid lipids, whereas oils are going to be liquid. And we'll talk here in a minute about what or why that happens. And then just some generic terms. So these are the three that we'll really concentrate on. So triglycerides, that's kind of the fat that we actually think about. So that's solid fat, or even the oils, those are mainly looking at triglycerides. But we'll also talk about phospholipids, which we've talked a little bit before when we talked about membranes, and same with sterols. So like cholesterol is an example of that. All right, so starting first with just what these are. So triglycerides, so the tri meaning 3. So these have 3 fatty acids that are bound to a molecule called glycerol. So 3 fatty acids per glycerol. And they are, those fatty acids are just long chains of carbons bound together. We'll see some pictures coming up here. So they can either have what's referred to as an alpha end, which makes them a little more acidic, or an omega end, so alpha, omega. So alpha being acidic, omega having a slightly different group called a methyl group. But mainly what we focus on as far as fatty acids is whether they are saturated or unsaturated. Saturated fatty acids are going to be solid. So these are from animal origins. Whenever we are making our triglycerides, our fats, they contain saturated fatty acids. However, whenever plants are making their triglycerides, they are using unsaturated fatty acids, and those are going to be more of our oils. So they're going to have liquid, or be more liquid at room temperature. And then both of them saturated, unsaturated can either have a cis conformation, which is what we want, but we'll also talk about trans fatty acids as well. All right, so looking first here at the saturated fatty acids. So again, there's an alpha end and an omega. So alpha beginning, omega at the end. So the omega end has that methyl group. Again, neither one really like water. But then this acidic group up here does allow it to attach to those glycerol molecules or to other molecules as well. So again, alpha end versus the omega end. But with the saturated fatty acids, what you see is they are completely surrounded or saturated with these hydrogens. So you don't see any double bonds here. So just single bonds between all the carbons in this backbone that makes up the saturated. So again, single bonds all along that backbone. Whereas with unsaturated fatty acids, along that backbone, you're going to have at least one double bond. So where these carbons are sharing 2 bonds with each other instead of only a single. So again, those double bonds, and anywhere that those double bonds occur, we get this kink in this tail. So saturated are straight, unsaturated are kinked, because everywhere that double bond is, we get a kink. And then we have monounsaturated, meaning there's a single double bond. But we also have polyunsaturated, meaning multiple. So in this case, there are 3 double bonds here. So this is a polyunsaturated fatty acid. So again, every place we're going to get a kink in the tail, and then where there are no double bonds, it's straight. still have that alpha and omega end, so omega at the end, alpha at the kind of the beginning where it can attach to that glycerol molecule. This is also referred to, so we'll talk about like omega-3, or maybe you've heard this term before. Omega-3 fatty acids, again, here's our omega tail or end. So if we go up 1, 2, 3, so it's that double bond is at the third carbon. There are also omega-6, so it would be the sixth carbon from the end. That's where those names are coming from. All right, and here's that omega-6. Again, here's our omega end. So if we count up 1, 2, 3, 4, 5, 6. So here is that double covalent bond at that sixth carbon from the omega end. So omega-3, omega-6 fatty acids, again, they have double covalent bonds, but we hear a lot about these. They're the best and some essential fatty acids that we need to be eating. All right, now the difference between the cis and the trans fatty acids, again, this is with the unsaturated where there are double bonds. What is the natural, normal, what our cells actually can make are referred to as cis, and that's where cis just meaning on the same side. So you can see the carbons here around that double covalent bond. They're both on the left-hand side of this double covalent bond here. So that's referred to as cis. And again, what happens with that is whenever the atoms align that way, that forms the kink in the tail. However, trans fatty acids are an artificial fatty acid, and in this case you see there's one carbon on the right and one on the left of our double covalent bond here. So trans just means on the other side, so they're on opposite sides. Now the significance of all of that, as you can see, whenever we have a trans fatty acid and those carbons line up on opposite sides, we have a straight line. So there's no longer the kink that we see on the cis. So it looks more like a saturated fatty acid. All right, so we have some essential fatty acids, just like essential nutrient is a nutrient we have to take in our diet. Essential fatty acids are ones we have to have from our diet. It's the only way to get them. We can't make them. And there are two of them that we have to get from our diet. Linoleic acid, which is one of the omega-6 fatty acids, and then alpha linolenic acid is an omega-3 fatty acid. So again, both of these we have to get from our diet. We can't make them. Now, once we have both of those, then the other ones can be made from those essential fatty acids. But we need those kind of precursors to make all the other ones that we need. All right, and this is just kind of showing that. So again, our alpha linolenic acid, again, it can be used and converted by multiple steps into this EPA, and then we can use it to make DHA, and then those can be used to make other fatty acids or to provide signaling within our cells. And then the same thing here with our linoleic acid. It can be used to make arachidonic acid, and then that is used to make others or to help with signaling or other things. But again, we can't make EPA, DHA, or arachidonic acid if we don't take those two essential fatty acids in our diet. So why do we need them? Again, they form body structures. So we need them. We don't necessarily use them by themselves, but we use them to make other molecules. They help with our immune function as well as our nervous system, and they can be regulatory. So it helps even like with our inflammation. They can be used in hormones. So again, but we have to take those in our diet. Right now, if we look at these triglycerides a little bit more, so again, triglyceride meaning there are 3 fatty acids, so 1, 2, and 3. You can see in this scenario, this fatty acid here, this is a saturated fatty acid. It's a straight line, no double bonds, whereas the other two, these are both unsaturated fatty acids. So they have these kinks in the tail where we have that double covalent bond. Right, so these 3 to make up our tri, they attach to this glycerol molecule here at the top. So we have glycerol plus 3 fatty acids. Now our phospholipids that help us make up our membranes, they also have the glycerol molecule that we just saw with our triglycerides. But instead of attaching to 3 fatty acids, this glycerol attaches to 1 over here. This is saturated in this case, and another. So there are 2 fatty acids attached to the glycerol here. Again, here's our kink, so we know that's unsaturated fatty acid. But then instead of attaching to the third one, it attaches to this phosphate group, which is then attached to something else. So that's the name of the phospho, because it has this phospholipid or this phosphate molecule attached to the glycerol instead of the third fatty acid. So we have 2 fatty acids in this phosphate group that then attaches to another molecule, in this example, choline. And we need those to help make our cell membranes. That's what creates that phospholipid bilayer to separate the inside and outside of our cells and to regulate what's coming in and out. Now our third class here, the sterols, so they don't have fatty acids at all. Instead, they have these 4 ring structures here. So those help make up what are referred to as sterols. They have these 4 fused rings. Again, they don't like water. And a couple examples of them, cholesterol is probably the main one we talk about. So this is found only though in animals. Other cells like plants or fungi, they don't make cholesterol. So this is only in animals that we see these. But again, they have this multiple ring structure. Phytosterols, those are found in plants. So again, they have these sterols. They're just a little bit different in our different cell types. All right, so just a little concept map to kind of put some of this all together. So again, our phospholipids here that help make our membranes. We have that glycerol with our phosphate kind of head region, and then these 2 fatty acids that are attached. Whereas triglycerides, we have that same glycerol molecule, but 3 fatty acids. And then our sterols don't have any fatty acids at all, but instead they have this ring structure. Again, that makes them not want to hang out with water and makes them in this lipid group. Now, as far as thinking about our triglycerides, we can, again, use them for energy. So we think about that 9 kilocalories per gram we get. So the triglycerides get broken down into the 3 fatty acids, and then those fatty acids can be broken down to release energy. And then the glycerol is also broken down. It can actually kind of act like a carbohydrate and give us energy as well. All right, so moving into section 5.2, looking at the differences between fats and oils and looking at them in our foods. So fatty acids and triglycerides in our food. So protein is where we kind of think about these and just giving some examples of where we find these triglycerides. So kind of some low-fat protein groups are things like kidney beans. Kind of a medium fat would be a loin or a round cut where there's not a lot of fat on it. Or then high fats would be nuts or marbled, just meaning like the fat's kind of embedded in it. And also processed meats, so like our lunch meats and that type of thing. Dairy also has triglycerides in it, so we think about low fat, consuming more of those like our skim milk or low fat yogurt instead of the high fat like the premium ice cream or even the medium fat like our whole milk or yogurt. All right, some other places we find them in the grains. So again, thinking about like our pastas and our breads, those kind of things. So again, low fat breads, cereals, just plain pasta. Kind of medium would be adding some butter to that bread or pie crust that has the shortening or the butter already in it. And then high fat would be that same pasta, which is low fat by itself, but then we add in the Alfredo that has the extra butter and a lot of butter in that sauce. Then also we can get some triglycerides from fruits and vegetables. So we think about our low, so most fruits and vegetables are very low fat. So they may have some triglycerides in them, but not a whole lot. Then we can move up to a medium with our baked potato, but we add that butter in to add in those extra fat or triglycerides. Then high fat avocados, actually we'll talk a little bit more about them, but avocados have kind of a larger amount of triglycerides in them, but they're a healthier kind of fat because they are unsaturated, but then are not so healthy fat would be French fries, where we're deep frying and adding in all of that extra oil to those. All right, so again, a little bit more here just about our avocados. So they are loaded with antioxidants, typically grown in tropical kind of warmer climates, or even California as far as the United States. Whenever you're shopping for them, what I refer to as Hass avocados are typically what you're going to see. So they're ripe whenever you can kind of are soft to the touch. But you want to stay away from them if they're dented in. That means they're probably overripe or have been bruised along the process. As far as storing them, once they're ripened, just at room temperature, and they can actually last for a few days or a little bit longer if you keep them in the refrigerator. As far as cooking them, again, the reason that even though they have a lot of fat in them, we call them good fats, is because they have those mono and unsaturated fatty acids, kind of gives them that creamy texture to them. Again, that's what helps improve their nutritional factors. They have a rather mild flavor, and then you can actually kind of use them as an edible bowl whenever you have that. If you kind of split it in half, it could be a bowl for like a salad or something like that. All right, so again, just kind of looking at where we get fats from, summarizing it here in our pretty little MyPlate tablet or table. So we have the fats and the oils just by themselves. So thinking about like butters, salad dressings, or just adding oil or cooking in oil. Again, some fruits, especially the avocados, but even berry pies. Now the berry itself doesn't have the 15 grams of fat in it, but the pie crust adds to that. So again, think about French fries. If we're frying our vegetables, that's when we're adding in all the grams of fat. Again, dairy, especially whole fat or whole milk, we can see some places there, whereas if we switch to the non-fat, we can decrease the fat intake there. Again, our breads and grains, we get some fat from them as well. And then the proteins, though, is typically is where we think about adding in those fats. So of course our steaks, but also the nuts, and then our fish, which have a lot of some of those healthier omega fatty acids, a little healthier version of consuming those fats. All right, so again, some of the food sources for those essential fatty acids, again, the omega-3, the alpha-linolenic acids, so places you can find these, so in flax seeds, walnuts have them, and then a lot of the oils, so like canola oil, soybean, perilla seed oil, and chia seed oil. We don't see those a whole lot. Probably the most one you're familiar with may be the canola oil here. So cooking with and frying with those provides that essential fatty acid. Then the linoleic acid, so the safflower oil, sunflower, also corn oil, probably the one, again, you're most familiar with there. Seeds and nuts, but also chicken has that fatty acid in it as well. All right, now the omega-3 fatty acids, we get a lot of them from fish, so it is recommended to consume about 2 servings of fish per week to provide that optimal cardiovascular health, just kind of showing, again, salmon has kind of the most amount of those fatty acids, but pretty much all of the fish, catfish probably having the least in there. So again, just some ideas whenever you are consuming different fish, which ones would provide those essential fatty acids for you. All right, then whenever we talk about those, just kind of consuming, again, the seafood twice a week, but also thinking about sustainability. So a lot of there's a lot of overfishing that has happened that's led to multiple, the seafood industry kind of being on alert, so they're not going extinct, but some of them are endangered. So it's just some ways to keep your seafood and get your fish in per week, but also not harming the industry. So using aquaculture, which is just fish farming, so getting those instead of getting them from the seas and the rivers, using those fish farms. So you can look for that aquaculture kind of late on the label to know that that's where they're coming from. As far as our phospholipids, so again, they help to make those cell membranes for us. So wheats, peanuts, our egg yolks have those, organ meats, soybeans, lecithin, but also they're used as what are referred to as an emulsifier to keep the liquid and the water mixture, so like salad dressings like that, from separating. So the emulsifiers, because again, oils and water don't mix, so emulsifiers get used to kind of blend everything together. All right, then the cholesterol too, just some ideas here of where we get a lot of cholesterol. So again, we need some amounts of cholesterol, but not too much cholesterol. So this is just showing kind of some where large amounts of that cholesterol is probably not something you have to really eliminate from your diet, but beef brains throws a whopping almost 3,000 milligrams of cholesterol. So again, kind of thinking that's not in your normal diet. But liver, beef liver, not a lot of people consume that, but it does have a lot of cholesterol. Of course, egg yolks have a lot of cholesterol in them. But again, cholesterol, we need some of it. We can't have too much, but staying away from eggs is not a good idea either. They provide a lot of benefits, protein and cholesterol as well. Shrimp can provide those. So again, just kind of looking down there, you can see multiple places where cholesterol comes into our diet. All right, so also whenever we think about fat and that satiety, so that feeling of satisfaction, making us feel full, satisfying those cravings, providing flavor and texture. So fat actually promotes that satisfaction because it's slow to digest, so it stays in our system longer, so it makes us feel fuller after a meal. Now, it does depend on the size and the saturation of the fatty acids that are in the food. So that varies from food to food. But it helps to improve the flavor and the texture. So especially like adding butter to things or kind of smoothing it out with a little bit of oil or butter or some other fats. So again, lots of flavor in there. But it can also increase our food intake because then it does taste so good. So then we eat a lot more of it. So we have to kind of balance all of that. All right, now it can. Fats don't last forever, so they can spoil, and that's referred to as becoming rancid. And I'm sure you probably smelled rancid fat, which smells absolutely disgusting. But what happens there, those oils do decompose. Now, oils, of course, can set out at room temperature. They last longer than our butters that have to be refrigerated. But that breakdown essentially of the double bonds and the unsaturated fatty acids, and it's mainly due to exposure to light and to oxygen. Now the polyunsaturated that have a lot more double bonds, they're more susceptible to that, so their shelf life is less than the monounsaturated. And then also, again, that rancid is really disgusting. It smells horrible, it tastes horrible. So that is what limits the shelf life. So there is an expiration on our oils, shortenings, that type of thing. So preventing that from happening, so adding different antioxidants to them can help. So during that processing, hydrogenated oils, adding hydrogen, switching them to getting rid of the double bond and switching them to saturated fatty acids. So decreasing light exposure, so using darker tinted glass or opaque packages so that the UV doesn't come in, and then decreasing the exposure to oxygen. So even using smaller containers so that you're not opening and closing it as much can help with that. And then also tropical oils are naturally saturated, so like coconut and palm kernels, so then they are not going to decompose because they don't have those double bonds to begin with. So then at home, some things that you can do is just storing those oils in cool, dark places so it keeps the heat and it keeps the light from breaking them down. So you can keep them in the pantry or even the refrigerator, even though it doesn't say to refrigerate them, just helps them to last a little bit longer. All right, so then our other one here, just kind of looking at how we break down these fats. So if we kind of a nice little summary table, we do start breaking fats down using that enzyme lipase that's in this in the mouth and our saliva. And then that's also going to be activated in the stomach whenever we get there. So the gastric lipase is released in the stomach. Again, it's going to help break down the lipids, hence the name. So it's the enzyme that breaks those down. Of course, our small intestine, that's where the majority of that's going to be. The bile comes in here from that's made in the liver, stored in the gallbladder. It emulsifies the lipids, again, just meaning it helps them to dissolve. So it surrounds them and makes them more compatible because they don't like water, kind of helps blend them into that. So that bile helps with that, as well as it has lipase that helps break those triglycerides down. And other enzymes from the pancreas and other accessory organs help as well. And again, the liver, of course, helps because it makes the bile. And then the pancreas helps by creating other lipases that help break those down by enzymes. And then by the time we get to the large intestine and the rectum, again, less than 5% of ingested fat remains, and that just gets excreted in the feces. All right, so again, just kind of putting that up in the slide. So again, in the mouth, we do have the lipase that's there, as well as just mechanically chewing and breaking apart the food to make it easier to digest. Again, in the stomach, that lipase continues to work. There's a lipase released by those cells in the stomach, and they're stable at acidic conditions. Then in the small intestine, the bile from the liver that was stored in the gallbladder, it helps to blend in and help dissolve the fats. And then the pancreatic lipase is sent to the small intestine to also help break those down. And so by the time we're done, we have these monoglycerides and fatty acids. So we're taking the glyceride off and we're releasing those individual fatty acids. Now, as far as breaking down phospholipids, because anything that has, especially plants or animals, they have cells, so they have a cell membrane. So those phospholipids are mainly broken down in the small intestine by enzymes from the pancreas. Again, we break down that glycerol molecule. The fatty acids are released just like with triglycerides. And then the remaining phosphorus parts also can be broken down further. And then the cholesterol, this is broken down by enzymes from the pancreas and the small intestine, so it doesn't start being broken down until we get there. And we end with cholesterol and free fatty acids. Right, and this is just kind of showing a nice little summary of this. So again, about 95% of dietary fat is absorbed, only about 5% stays in and is released in the feces. So again, it's a product of digestion, diffuses into those absorptive cells that are in the small intestine. So diffusion means that they're just moving across, because again, they're unlike amino acids or carbohydrates, they don't like water. So they can cross through the phospholipid bilayer of our cell membrane. So they can just go from a high to low concentration, but they do need to be small. So they're broken down into short and medium chain that can be absorbed, and then they go through that hepatic portal vein. Now the long chain, the bigger ones, those are going to the ones that are absorbed by the lymphatic system. That's one of the jobs of the lymph, because it's kind of sampling the fluid around the cells. So it's going to take those larger fat molecules. And our picture here is just showing, again, a large fat droplet coming in, has lots of triglycerides. So the bile is going to combine with it, kind of sticks these things in it that make it get along with water a little bit better. So then the lipase is released. That's going to break those down. So we're going to get individual fatty acids. We're going to get some longer fatty acids that are in there. So again, the monoglycerides and can just diffuse across. The longer chains have to go through the lymph first. Once those short fatty acids get in, they can combine with the glycerol and they actually make triglycerides again. So then that goes into the absorptive cell. They then get surrounded by what are referred to as chylomicrons, which we'll talk more about later. And then ultimately, because they're so large, they then enter the lymph system, but ultimately get into the cardiovascular. Now, those short and medium chain fatty acids, they're actually going to hop right on the portal vein and go straight to the liver.